All right, let's dive right into the hidden mechanics of epidemiology. We hear about outbreaks, viruses, and public health all the time, right? But the underlying science, the actual nuts and bolts of how a pathogen moves through a population, is a seriously gripping puzzle. Today, we're going to unpack the hidden rules of viral spread, and we're relying strictly on the data and the models used by the professionals who track them down.
So, to kick things off, consider this. What do a leaky bathtub, an Asian tiger mosquito, and your ancient Neanderthal ancestors all have in common? Sounds like the setup to a really weird joke, doesn't it? But as we'll see, these three totally unrelated things represent the fundamental concepts, the environmental shifts, and the hidden biological triggers that dictate how diseases spread across the globe. We're going to solve this mystery together today.
Well, to figure this out, we've got to meet the folks actually doing the legwork, epidemiologists. Think of them as the elite disease detectives of the science world. They are constantly on the hunt for microscopic clues, working everywhere from laboratories to state health departments to solve global outbreaks before they spiral totally out of control. What exactly is in the toolkit of these investigators? Usually, armed with a master's degree in public health, their mission is actually pretty laser-focused. They have three main jobs: hunt down the root cause of an outbreak, pinpoint exactly who's most at risk, and ultimately figure out how to stop or prevent the spread. It's high-stakes detective work where the culprit is completely invisible. Okay, so how do they actually measure all this? They use something called the bathtub analogy. It's super simple, but honestly, it's brilliant. When trying to measure the presence of a disease, epidemiologists use this visual framework. That tub right there, that represents our community. Now, think about the water dripping in from the faucet. That's what we call incidence, the brand new cases dropping into the community at a specific point in time. The water that's already sitting there in the tub, that's prevalence, or the total existing infected individuals. And just to finish off the metaphor, water evaporating is people recovering, while water going down the drain, well, that represents mortality. It really gives you a perfect mental picture of how an outbreak flows. So naturally, the biggest question is just how fast is that faucet running? That's where we get to the basic reproductive number, or as you've probably heard it called, R naught. This is simply the average number of people who will catch a disease from just one contagious person. But keep in mind, this model makes one huge assumption. It assumes a completely susceptible population. That means absolutely zero previous immunity. Nobody has antibodies yet.
And the math here gets pretty terrifying pretty fast. It all starts with a single patient zero, creating this exponential branching network of infections. Whether that R naught number is high or low basically comes down to three things: how long the person is infectious, how many vulnerable people they bump into, and how the bug actually travels. Like airborne viruses, they generally have a way higher R naught than ones spread through bodily fluids.
But hey, math on paper doesn't always survive contact with the real world, right? Enter the wild card, super spreaders. Have you ever heard of the 80-20 rule? In epidemiology, it means roughly 80% of new transmissions are often caused by just 20% of infected people. You drop one contagious person into a massive packed crowd, a super spreader event, and suddenly all that standard R naught math goes completely out the window. That bathtub floods fast. All right, let's shift gears. We know how fast it spreads, but how dangerous is it? Think of this like a funnel. At the very top, we have incidence, all the new infections. But not everyone gets visibly sick, right? So we filter down to morbidity. Those are the folks actually showing signs and symptoms. And then, tragically, we funnel down to mortality, the fraction of the population that actually dies from the disease. To put a hard number on that lethality, our disease detectives use the case fatality ratio, or CFR. Once someone is officially confirmed as infected, the CFR tells us what proportion of those people will ultimately die from it. So if R naught tells us how contagious the suspect is, CFR tells us how deadly it is once it gets in the door. And what's really fascinating is the tension between those two numbers. Usually, highly lethal viruses, think Ebola or bird flu, they have massive fatality rates, but they don't spread all that fast. On the flip side, measles spreads literally like wildfire, but has a lower fatality rate. Then you look at the new coronavirus, and it sits right in this unique, highly impactful middle ground. But here's the catch. a single CFR number can be a totally misleading clue. Lethality is almost never just a flat rate. If you look at COVID-19 data, the case-fatality ratio shifts massively depending on demographics. For kids aged 0 to 9, it's a microscopic 0.002%, but for adults over 80, it skyrockets to 13.4%. So disease detectives can never just treat an entire population like one uniform blob. Section 2. The unexpected suspects. Hidden factors of viral spread. Okay, so we've got the basic tools down. Let's zoom out to the macro level and look at the unexpected parameters operating behind the scenes. Remember that Asian tiger mosquito from our opening puzzle? Well, as mean temperatures keep rising across Europe, the geographic range of these bugs expands drastically. Climate change is literally acting as an active accomplice here, moving carriers of viruses like chikungunya into entirely new communities that have never faced them before.
And it is definitely not just mosquitoes. Changing up our environment has a profound effect on zoonotic carriers, animals that can pass diseases to us humans. Shifting vegetation and habitats over the past century have forced diverse bat species to migrate into new global hotspots. And by doing so, they're bringing hundreds of bat-borne coronaviruses closer to dense human populations, basically laying the groundwork for the next patient zero.
So those are the external factors. But what about the internal ones? This is where the case takes a huge twist. It turns out specific genetic risk variants, allele frequencies, are strongly tied to severe COVID-19 reactions. And guess where this genetic risk comes from? Chromosome 3, inherited directly from our Neanderthal ancestors. Crazy, right? Literally, your ancient genetics can dictate the severity of a modern virus. And these Neanderthal DNA variants aren't distributed evenly at all. They're carried by roughly 50% of people in South Asia, compared to only about 16% in Europe. That ancient biological legacy creates a wildly uneven playing field, where an entire region might be statistically more vulnerable just because of DNA inherited tens of thousands of years ago. And beyond ancient DNA, there's one last biological clue we need to talk about: biological sex. It plays a massive underlying role in viral spread and severity. See, immune cells actually express sex steroid receptors, and those regulate gene expression. Because of this, biological females generally exhibit higher immune activation, while biological males exhibit lower immune activation. It means our disease detectives absolutely have to factor in biological sex when they're mapping out the morbidity and mortality of an outbreak. So let's pull it all together. We've connected the leaky bathtub of community spread, the climate-driven migration of the tiger mosquito, and the hidden vulnerabilities buried deep in our Neanderthal DNA. When you take the strict math of the basic reproductive number and throw in shifting climates, global travel, and our own genetic variations, the puzzle becomes infinitely complex, which leaves us with one final, pretty provocative thought to consider. With all these evolving factors constantly in the mix, where will the disease detectives be needed next?